Here's our packet. This is
Come on, it's fun. All right, so we'll get started. This is big idea number one. What is big idea number one? Evolution, right? So we're going to talk about the 3 labs that you may or may not have completed on evolution. My name is Dr. Kathleen Ireland. All my students call me Dr. Kate, so feel free to call me that. I teach in Pensacola. I teach at Pensacola State College right now. I taught high school on Maui for 25 years before that. So I'm pretty well versed in the high school situation, and now I'm into college, which is a whole new ballgame. All right. No idea how to work this computer. There we go. Okay, so the point behind this, I'm a math person, I don't even know what that is. So the point behind this is that we're going to help you get through the FRQs, the free response questions, when you go to take an exam. I've been working with the AP College board for more longer than probably your parents have been stuck in there. So I've seen everything that can happen when you get to these FRQs. Here's what people always stumble on. What is it we're asking you to write? So before you even do anything with the free response question, you should read it. And I suggest reading it without any pens or pencils in your hand. Read all the questions. You've got 10 minutes before they begin timing you for how long it takes to do the answers. Use that 10 minutes. Read the questions. Second, pick up your pen, read them again. This time, jot down any words that come to your head. It doesn't matter if they fit with it or not, just write them down. We don't grade anything above the question area, so you can write all over that question, and it will not detract from your grade in the least. So write anything you want. Write any word that pops into your head, because probably will have something to do with that. The last time, go through and look for the sequence of what we're asking. It is so much easier to earn points if your points follow the sequence of the question. When we get into grading these exams, it's a huge room. It looks like your gymnasium. It's broken into 4 areas. We read one group of questions, and that's all we read for the entire week. And we read them thousands, literally. Last year there was 186,000 exams to grade. 186,000. There's probably 400 to 450 teachers grading those exams. So the easier it is for us to find your points, the more points you will get. We have to go through them all in 4 and 5 days. So it's helpful to us. If you can write the sequence that we're asking you for, and then you put a quick outline so that when you get back to that, you can answer that question. Totally untrainable. Okay, so when we come to the FRQs that we're going to work with today, these are the terms that are on our FRQs today. Identify, justify, describe, predict, explain, and construct. Do we know what those mean? When it says identify, what am I asking you to do? Figure out what we're asking, and then what? Yeah, I want you to point to, or like, if you could point to it, I want you to write down exactly what the relevant information is. Identify is a pretty easy point. It's almost like a vocab. Give me the answer. Give me the vocabulary. What about justify? I ask you to justify. Defend your answer, right? Your answer, most times when we ask you to justify, we ask you to give us a choice or an opinion. And then we're looking to see if you can use the scientific terms to give me the reason why you did that. Justification points are pretty easy to get. As long as your justification makes sense and is logical, they're pretty easy to get. Oops, describe is just to give me a detailed listing, right? What about predict? When it says predict. Yeah, this is the easiest point on the planet. Predict is tell me anything. Just give me some random prediction. One year we had a test question on celery seeds and germination with and without sunlight and what would happen. And one of the students' prediction was that the celery seeds in the dark would hatch dinosaur eggs. Okay. There's a point, that's a prediction, and then they tried to justify but did not get the justification point because it was pretty obvious that it was very difficult to justify that. But they did get an easy point. So predict, if there's a predict, you can, I got at least a point on this one. What about explain? Elaborate, tell me everything you know. Step through the process. It's usually explain the phenomenon, explain the process. And the last one, construct. Construct is draw. Draw a graph, diagram a procedure, lay out the steps. Okay. So those are the terms that we're going to deal with today, and if those were the terms that you had on your FRQ, when you get to the questions, you should, in the top portion on your second read-through, jot down really quickly what you're going to do with those. Especially if it's predict, you just circle it and star it. That's what I was saying. Okay, so as you're working through this, what if nothing comes to mind? What if you read a question and you're like, I don't even know if this is English. I don't know what to do here. What are you going to do? This happens. I've seen this happen.
Yes.
First, huh?
Yes.
Yeah, well, don't guess. The first time that you read through it, if nothing happens and you've never seen these words in your life, move on, because there are 10 free response questions. So move on. And then you come back to it the second time. If you're done with the pre-read, think about the big 4 topics. What big 4 topic might this be talking about? I have no idea what those individual words mean, but maybe this is an evolution one, so I'm going to write evolution on top, because I know a lot about evolution. So I'm going to write that on there. If you can identify then, did I do a lab or anything that seems similar to this? Write that down up there. You underline any terms you recognize, you can at least define those terms, and maybe that'll help you get a point or two. If there's a graph or an illustration, undoubtedly you can get a point just by explaining what that graph or illustration says. So even if you have no idea what they're asking, you can grab a couple points. Never leave it blank unless you run out of time. Writing a story about the prom is entertaining for the person who's grading it, and we do have to read the whole thing through, but in case you stick a biology term in there somewhere, but it usually doesn't get you any points. But throwing out some vocabulary that you see in there, talking about the illustration, talking about the graph, you might be able to pull a point. And the final thing, every year that I've worked on this, every single year, I was question leader, which is in charge of a group of questions, 3, 4, 5, or 6, 7, 8 for the last few years. Every single time I've done that, one of those 3 questions has been so bad. The average is down here, like out of 3 points, half a point is the average. Right? So when you get to one that you don't know, don't think you're the only person on the planet that doesn't know that. Chances are that's the question this year that's going to be terrible. So again, anything that you can identify up there, anything at all, will get you a point, which will put you above the average, where people go, oh, I don't know, and move on. So don't give up. That's what I want to say. Don't give up. We every year, and sometimes it's the big 10 pointer, the 1 or 2, number 1 or number 2, the big 10 pointer that just, one year we had graphs of mice. Yeah, it was tough. So don't panic, you're not the only one. All right? All right, let's see what we can do with this stuff. So this time we are going to do something that I've not done before. You see you're in groups of 1s, 2s, and 3s. We're going to work in our little groups. The first thing we need to do is figure out if we know the vocabulary. So if you open your packet, there is a listing of Strive for a 5 vocabulary. Those are the words that we're going to see. So go ahead and look at those for a minute, and then we're going to play a game with them. Okay, hopefully these are words you know. You're going to pick a partner. In the tables where there are 3, we'll have one group of 2 and one group of 3. One person, partner A, will face the screen, and partner B will face the other direction. In this case, B and C will face the other direction. And I don't know if you know, I'm really old. They used to have this game on television called Password, where one person would give all these clues, but you can't say rhymes with, you can't say starts with, you can't say sounds like. It has to be actual clues for the term, and the other person tries to give them the term. That's what we're going to do here. You have a minute, and then we'll switch places, and we'll do it with the next couple words, and then we'll switch places, and we'll do it again. This will do 2 things. It'll remind us of the vocabulary, and it'll let me know where we are. Okay? So go ahead and get yourself organized so that one of you is facing the board, and one of you is facing that person, in this case 2, and in this case 2. Oh. Are we good? You'll remember it by the time we're done, I promise. Okay, I don't know how to set a stopwatch on my clock. All right, so we got one person facing the board and the other 1 or 2 are away from it. You have 1 minute. Ready, begin. Okay, 10 seconds. And stop. How'd you do? 2, that's not bad. You'll get there. Okay, mark down the ones that you got, and then switch places. So the other person, or one of the other 2, is giving the clues.
No, he's looking this way. We're going yeah.
Ready? Okay, here comes the next set.
We talked about the first one. How is it about? Like vibrating.
10 seconds. Heterozygote advantage is, I'm sure your teachers talked to you about sickle cell anemia, where the heterozygote survives malaria. The homozygous recessive gets sickle cell, that's a problem. The homozygous dominant has normal blood that malaria loves. So the heterozygote has the advantage in that population. Okay? All right, switch places.
Are we still doing 4?
Yeah, the last one is 5. Okay, we ready? And go. Oh, these are easy.
That would cause a no, it's a bad thing. Yes. Okay, say the gene.
Oh, yes, so you can say
Fall out.
It's a certain trait that something would have.
It's not drift. Not drifting. Is it no, it's not.
We're coming in and out of the population.
You think of brain with this.
Oh. You know about animals.
Also shows it's like
I don't know what it is.
I don't know.
I know that those
5 seconds. All right. How'd we do? We know these terms.
We just cladogram.
Cladogram. You know what a cladogram is?
It looks like a tree, and it
Yeah.
has one big bar, and it has a bunch of little bars.
Exactly. It looks like a tree with one big bar and a bunch of little bars and derived characters. Yeah. All right. Switch again.
Wait, is this me again? Oh, thank you. Oh, okay.
Switch one more time. They switch places like physically. Ready? Go.
Yes, but
I think this is
Ten seconds. And stop. Okay, last one. So we do all right? Yeah? So now we remember all the terms. We know what we're doing for today. Yeah? All right. Have you covered the Hardy-Weinberg principle in lecture and lab? Good deal. So the rest of this, I know this is really small, but this is how today is going to work. These are groups 1, A, and B. Groups 2, A, and B. Groups 3, A, and B. Each of you has on your desk a paper clipped together slides. These are also in your packet. These are what you're going to work on at the group. And we all have a poster board. So your job is going to be, I'm going to give you 10 minutes, and I'm going to walk around and make sure everybody's on track and we're all doing the right thing. You're going to work on part 1, part 2, or part 3. When you do that, we're going to leave the poster in 10 minutes. You're just going to move. 1A goes to 2A, 1B to 2B. You guys go to the 3s, and the 3s walk all the way around to the 1s. 10 minutes, and then we'll do it again. The posters stay where the group is, and we add to the poster each time. So by the time we finish, we'll have everything we need to know on the poster, and we will be rolling. Are you confused? Yes. Okay.
what are we putting on?
I will show you.
Okay, all right.
It's okay. After 30 minutes, we'll make sure everything's done. We'll make sure everyone's pages in their booklet are filled out perfectly. If there's any time left, we'll play another game, but I don't think there will be. And that's what we're going to do today. So here's how it works. 1A and 1B, this is you. First, you're going to make a concept map. You know what a concept map is? A concept map, in your case, is going to look like this. It's going to say artificial selection. And natural selection. So those are your two terms. Concept maps are anything that relates to those terms. I would like you to put at least one for something that is similar between the two, and that would go here. What's something similar between artificial and natural selection? Okay, heredity. There, that's good. Now you already have one. Then I would like you to put at least one difference between them. And then the next group will add to that. So that by the time we're done, we'll have at least 3 similarities and 3 differences for each one. So you guys will have a hard time. But that's okay. It's okay, I'll help you out. When you finish with your concept map, so you get to set it up in the beginning. When you finish with the concept map, you're going to choose from your packet that's staple or there. From your packet with the paper clip, there are 3 student samples. So you'll read the question that's in your handout, and then you'll read one of the 3 student answers. And you're going to decide, is that a good one, a bad one, or an awful one? And you're going to look at it and determine what is good, bad, or not so good about that. Talk to one another about it. Write down on your chart what you see or don't see in that particular response. And finally, page 3 of your handout, there is a table. This is the table of construct an experiment. So there's some information on that table, and your job is to complete that so that you can construct an experiment that will work perfectly. You don't have to write the experiment, you just have to figure out what you would put on it. That's you guys, and everybody else when you get to that stage. There's the concept map blank, there's the blank for the student samples, and there's the blank for the artificial selection design and experiment. Middle tables, 2A and B, you guys are Hardy-Weinberg. Top of the page 3 of your handout, there are some questions for you to answer, and then on the top half of your poster, you're going to write the 5 conditions that are required for Hardy-Weinberg equilibrium. If you don't know all 5, that's okay, I'll help you out. The rest of you, when you get there, will just make sure they're correct and they make sense to you. And then you're going to complete the table on page 4. You're going to say what equation you use, and you're going to go ahead and work out the phenotypes. On the bottom half of the poster, you're going to write some conditions that would alter Hardy-Weinberg. So I'll help you brainstorm some conditions that would alter the Hardy-Weinberg. And then on page 5 of your handout, part C says pick one and tell what would happen. Okay? So that's what you guys are going to do in the middle. This is the first and second, and this is the third thing that you will fill out. And 3A and B are doing BLAST. Y'all did the BLAST lab? Nope. Do you know that as organisms diverge from one another a longer and longer period of time between the common ancestor and the organisms, their DNA changes increase? That's what the whole BLAST lab is about. How many DNA changes do we see, and what does that indicate as far as making a cladogram? So in your handout on page 5, there's a bunch of DNA strands printed out. You will analyze those and answer the questions. And then there is a table that shows you the differences in milk components between cats, cows, horses, humans, and pigs, because they're all mammals, right? They all make milk. So there are differences in the milk components there, and you're going to look at that and draw a phylogenetic tree. On the poster, by the time we're finished, there will be 3 phylogenetic trees, and you can draw it as a cladogram, the big line and the little ones coming off. You can draw it as a tree, the football brackets. You can do whatever makes sense to you, and then we'll make sure that it's justified by the time we're done. And then you have the final question. So yours, here are the DNA sequences, and I will show you if you're not sure how to do that, I'll show you how to get through those. And then here is a cladogram, and yours has the footballs in it. Okay, That's what we're doing. Are we good? 10 minutes to start the first one where you chose to sit, and then we'll move forward. Okay, I will come around and help you out. So you guys are working on your concept map. You guys are working on Hardy-Weinberg and what are those equations. And you guys are staring How are we doing over here? Yeah, you know what those equations are for? Okay, so the P and the Q, what are P and Q? Dominant and recessive alleles, and the frequency of those two alleles has to end up to be 1. So the first one is the allele frequency. What's P squared to Q and Q squared? Oh, sorry. That is the equation. What are those numbers? What do they mean?
To find out the percentage of.
Yes, the percentage of homozygous dominant, homozygous recessive, and heterozygotes. So that gives you percentage of, not exactly phenotypes, because dominant and heterozygous are the same. Percentage of homozygous and heterozygous individuals. How many? Well, these go on your, I don't know. Most of you have 5 equations. And then, so you'll fill this out right here while you post work. You don't have to put gross.
Okay, so now we're talking about like how the selection of how they, yes, how to protect themselves against rather than just how they evolve and
Good. The poster I want you to write artificial selection and natural selection. Yep, and that's the only thing. Okay, so you'll have 3 terms here, and then everyone else will
try
Okay, so once you finish this, where it says, this is the question. And then you choose one of those to look at. Just write down which one you choose. And you'll say, well, should they do well? That's the number of pairs. Okay? How we doing, guys? So do we have something to put on there?
like the selection of plants in there, they're protect them against variation.
Okay. This, yep, that's exactly right. This was. But anyway, that's exactly right. That's what the question is about. So here you want to put down, before you even get into the question, you want to put down one thing that natural selection and artificial selection have in common. Not, just in general, not thorns and plants, just in general. Yeah, they're both pressure on genes. So just, that goes in the middle, right? The circles that go between both. Okay. And that, no, I don't even know why they made me copy those. Make sure you copy those. And then you need to tell me one thing that's different, how artificial selection is different, and how natural selection is different. Okay? And then you're done with the poster. You don't know the 5 conditions for Hardy Weinberg? That's okay. That's okay. You know that in order to be in Hardy Weinberg equilibrium, that population is not false. And that never happens. That's what Hardy Weinberg is all about. And how much evolution is happening in this population. So if you're having a Hardy Weinberg population, Hardy Weinberg equilibrium, no things happening. The 5 things that are required in order for Hardy Weinberg, in order for no evolution are no mutations, no DNA, your billion base pairs, they mutate one at
Will this spell for natural selection? Darwinism?
large population. It has to be a large population, small populations. No gene flow. So you got this non-mutating locus population with no one going to 15 random mating. You can't select based on, you have to just mate blindly, no selections. And the 5th one. Gene flow random mating, no mutations. Throwing a huge blank.
So many things I didn't know. Like, what is it?
Have you done
Oh, yeah, exam. I'm sitting class like.
Oh God.
Yo, coach.
I wrote them down so I wouldn't do this, and now I can't find them either.
Useful.
I'll come back. Go ahead and use the other 2. Hey, how are you doing? Oh, that's it. No natural selection. No selection. Thank you. All right. So let's go here. There's one more that's so simple. So no selection.
I did. You know, she's like a. Hey, Laura, you should have seen everybody. I know you.
So you can put either one. All right, are we doing better now? Nice. Okay, and so no natural selection means that no gene is, no allele is better or worse than any. How are we doing down here? Wonderful? We're done? Cool. We are wonderful. Let me see what you got. Oh yeah, you're supposed to do all at once. You got 10 minutes. All of one. How are we doing now, ladies?
Oh, Jesus Christ. That's why she wants to
Very nice graph.
No, but.
Like I said, here's how
I have to read the student samples. I need to like explain. You good? I can't read it.
and I have my cursor here.
By type of cursive. I used to write cursive. Z.
It feels like I have a lump.
How are we doing?
Check yourself.
Okay. So now we have to calculate a little bit.
Yeah, I think we have to do that.
Okay, we're on.
Is it how many more?
No.
Does this happen? It can't. No, some of these can happen. So the heart tells us how much of the population.
Don't something do overlap unsignificantly.
No, I would just.
Get in the red bar that passed behind the sector.
Yes, sir.
Alright, this one I'm not using
Okay,
now
on your
because I
report,
can
you're gonna write the 5 conditions that I gave you. Just write them, and then you're gonna write your environmental brainstorm. What can change? What factors can affect?
You mean explain this one? I just couldn't read one. No, you did answer basic. He did basic.
Okay, what about that graph? What is that graph?
That looks like Hardy Weinberg. No.
You write, yeah, the cloud condition.
Is that the right graph for that particular experiment?
You can just check it without equation.
What is that graph? I mean, I'm asking,
Oh, he did, but he is not the right graph.
it's not complete. I don't think that graph is complete. I see a dot and some error bars. But wouldn't there be an easy way? Look at your other samples. Oh, what? Yeah, that's simple. Points all over the board. That one's tough.
I mean, I know what he did.
Yeah.
I just forgot what this graph is called.
That's looks like a stem leaf. That's okay. You don't have to know what it is, you just have to know that you have the right graph. There's an easy Yeah. This one did earn all 10 points.
Wait, what are you?
Yes, but not for the graph. There's always, if it's 3 points to a section, you can get an explanation point. So there's always there's always 12 points on a 10-point. That's the possibility.
And then you will say that it's not a large population.
You doing all right? Yeah. All right. So the other thing that goes on there are all of the brainstorming environment. We got we have a couple more minutes on the stations. Are we doing okay? Feel okay about natural and artificial selection? That question was a tough one. The average on that out of 10 points was 3 and 4. I feel like you got it way ahead of me.
Yeah, he did make it. We made it hard right
On the next page.
right here.
I think I'm gonna switch us pretty quickly here, and that's okay that you didn't get done. It's okay. Yeah, go ahead and start. That's great. How are you doing this? Okay. Yes, that's excellent.
Yes, I have.
Write that on your ring.
Great. 6 o'clock. So I had to be or once you do it for so like me, I got the call.
Got you. Is this good? Alright. Okay, we're gonna go ahead and just write. No, just this. Just Can you finish that? Okay, go ahead and put the caps back on the pens. Change from, we're all moving this way, you guys are moving all the way around. It won't take as long for the second and third groups because you already have some things on the poster. Alright, so now group 1, you're going to add to the already begun concept map. And then you're going to move forward by choosing an essay to read. Group 2, you are going to make sure the Hardy-Weinberg equilibrium conditions are correct and spelled correctly. And then you are going to work on the figuring out the equations and then the environmental change. Group 3, you are going to first do the last portion, which is find the differences in the DNAs. And then you're going to work on the Let me show you. done this before? Okay, okay, so what that means, those are the actual ACT and G sequences. Your job is to find the course. I do all the ACT. There's computers already. When you get all the way done, you're going to count all the differences, and that's how you show different relationships between them. No differences, very close.
I think
Lots of differences, you and a fish.
yeah, we
You know and you know that the more differences there are,
If you look at everyone else compared to
because he is, I mean, it's really different.
they're already different.
They're not lined up.
All of them. Find them. Wait, you're just gonna look at them all as first we okay, so anything is different. Yeah, I would like that. I'm coming. Okay.
There's two A's and two B's.
as indicated in the table.
Make that true. How about pressure on
It's
that is what it is. Sometimes I carry a lot of mutations with this pressure, but then maybe something will change and they will become real one.
There's a lot of money. Is this a show? Yes.
Okay, what's up?
Just non-random, right?
Artificial selection is non-random. Well,
I mean, to a certain extent, but
in some organisms, like sea urchins, you know, they just throw eggs and sperm into the sea and say good luck. That's random. But in most vertebrates, it's not random. So yeah, I would write vertebrate or something. How are we doing? Hard? So okay, so here's where we are.
What do we use this question?
Yeah, population is sexually dominant, yeah, frequently dominant.
Yeah. So this one's gonna tell you all random. So that's it.
When you get like 0.4, yeah, chunk factor percents of new chunk factor percents,
And what that is
the dominant, recessive.
Wow. I check off one day.
Heterozygous. Homozygous, heterozygous.
This is
Just right here. That's not how that goes. Actually,
I'll check out.
Oh, are we good now?
yes.
I'm gonna pull this sample.
All that to make one 5% is just what each one makes up. It was inside the field, inside the bottle.
That's okay. So that's what I, all right. All right, this is working now.
That's something. Just able to
Are we good with this one?
Or just keep this and look at this and have done better.
No, you didn't do all this stuff. This stuff. This stuff. What happened? Oh, I see. This is a math class. They have to have one.
have this one is too short.
Now since they have a nice big playground, it makes sense to you.
It works. X and Y.
But no mutations. Large population.
Pretty cool, huh?
Now they like that ball get the ball neck and I'm like random short half.
Are we good? Okay, we have only 7 minutes left, according to this clock. So here's what we're going to do. We're going to finish up where we are now, and then we're going to talk through each one of them. So the one that you didn't get to, we will at least be able to fill in, okay? So let's do this 2 more minutes.
We still don't know what's going forward.
Okay, what's 4 seconds?
They just don't. They kind of just give me a paper like, oh, ask her. Alright.
Alright, let's see what we got. So, we're on this one. Oh, okay, so we're now here. What last group thought was a good representation of the changes in milk proteins based on those factors. So we look at this here, we can see that all of them have common ancestry line.
Think tomorrow, Saturday.
Everybody has that. Everybody has protein A. So they're on this too.
You tell this legit, like legit. You would have 10.
They all have protein A and C. But the human has.
Yeah, 'cause look, I picked up C and I was thinking about all the plays of music and I saw her snap and I was like, I bet you she needs a ride. Yeah. Like, I bet you she needs a ride. Me and Z were jamming out one way.
Okay. Let's look at.
I'm gonna pull this massage. Maybe we get a great massage.
Okay, so here are 2 for artificial versus natural selection. What is artificial selection? People determine, right? I have a beautiful black lab puppy. Beautiful. Artificial selection. My neighbors have a rescued mutt, which is actually a much nicer pet. Natural selection. Okay. So artificial is people choosing. They both have to do with heredity. They both have sort of non-random mating, especially if you put it with vertebrates. Right? Vertebrates generally they have selective mating. But some of the inverts have random mating. They throw their sperm and eggs into the water and walk away, and it's random. So that's pretty good. Natural selection does occur naturally. It puts pressure on random mutations in the DNA. Artificial selection we choose, and it occurs in a controlled environment. So those are good. Does that make sense? Okay, this one, teeny tiny. Most useful to humans, artificial selection, that's true. Pressure on genes for both of them, changes in allele frequency. Natural selection is Darwinism and survival of the fittest. So those are good. The next part of that, you were to look at a sample of writing, and the hope there, sorry, the hope there was that you would notice that a bar graph is acceptable and easy and well labeled. You need units, you need to label them, and that it was in nice, even order. The three examples there were a 10, an 8, and a 6, so you didn't have any of the really bads. Yeah, that yours was a 10, the one that you guys were looking at. The one that you looked at was an 8. So that tells you a little bit about how those points are earned. Your goal there was to notice that you need to write logically, and a little bit is good. You don't have to fill up that whole thing. A little bit is good. Okay, the next part of that one was a table that was basically how do you set up an experiment. The first column says independent variable. The summary is write the units, right? Independent variable, write the units on the graph. Don't assume I know anything. Write the units. And always remember the independent variable is the one that you're, which one is the one you measure, one you change? Which one is that? Change is independent, right? One you change. The one you measure is the dependent. And again, units. You can't just assume that we know what's going on. So units. What's the point of the control? Have a difference.
Good morning, students. Thank you so much for showing up here on a Saturday morning. The following students have won gift cards. If you'll report to the Live Oak Pie table, we will issue you those gift cards. Congratulations to Caitlin Todd, Madison Swearingen, William Tryon, Taylor Miller, Alexa Payne, Morgan Jones, Max Brown, and Anthony Shade.
We're going to put out new poster boards, so if you want to help me by taking your
And at this time, we'll rotate to your next
sure that
session.
everything else is still on the table there. Just